Good morning. Thank you for joining us bright and early. I know it's going into another busy day. Hope day 2 is looking good. I'm Adam Roose. I'm the moderator today for this panel on the acceleration of the development of cell and gene therapies through the use of machine learning technologies. Got a great set of panelists today who are leading thinkers in the intersection of these two technology industries. I'll give a brief introduction to myself and then turn it over to the panelists who will make much more illuminated remarks than I can. I'm Adam Roose. I'm the VP of Cell and Gene Commercial Strategy at Adjuvant Partners. We are a boutique consulting practice focused on developing trends in technology, primarily focused on cell and gene therapies, but increasingly in the machine learning space. As part of that work, I'm the co-founder of the Alliance for Artificial Intelligence in Healthcare, which works to solve and resolve common challenges across the industry and the sector around the development and implementation of machine learning in healthcare systems. Excuse me. So the panel today is going to focus on the way in which cell and gene therapies can be affected by machine learning, or the way that the technologies can be applied and improved through machine learning to accelerate development of therapies. Excuse me. I'm going to turn it over to Bob to introduce himself, and I've asked the panelists to introduce themselves as well as their companies and the way in which they're combining cell and gene therapy technologies with machine learning to solve current challenges and improve program efficiencies. So Bob.
Yeah, great. I'm Bob Deans, CSO at Synthego, and contrary to Chris's point, I'm not a rookie. I have been here before. So I'm real happy to be here, actually. My past has primarily been in the life sciences sector, in the biotech industry, working on a number of programs, including iPSC while I was at Blue Rock Therapeutics, engineering HSC when I was at Rubius Therapeutics, and a past emphasizing CMC and development processes. So I was real attracted to Synthego because it's an engineering company. And the founders of Synthego were two software engineers that came out of SpaceX, and at the time had a glimpse of where we were around 2017 in terms of the CRISPR industry, and realized that there was a need to produce synthetic guides to be used with the nucleases to sort of open the research market in genome engineering. And so they built a novel solid-state synthesizer to make guides cheap and quickly, and were able to seed the research market, which at the time was really exciting, a really innovative space for CRISPR around 2018 or 19. So they realized that people were buying the guides for purpose, to edit cells. And so they also stepped up to produce the ability to generate automated robotic platforms that could take synthetic guides and very reproducibly edit cells. So it's really taking engineering principles and applying them to biology and building out a platform to have high reliability and quantitation for the products that were made. And so the session here is really around how ML or AI or machine learning can be used to augment therapeutics in the cell and gene therapy space. And so one of the things we realized is that having built the guide business and the ability to make the guides, having built an automated process to refine and accurately edit cells, we could also step up and build automated platforms suitable for GMP. And we realized that in the process of starting with guides and going through the food chain, we had built automated engineering platforms that could do target discovery, could do process development, and now we're planning to be able to do GMP cell editing. So really building out like a Dunkin' Donuts kind of franchise for a genome engineering company. And so we use machine learning in a number of ways there. In the guide chemistry business, we're heavily focused on all of our operating parameters in first pass yield and quality components, and we use machine learning to crunch our data set. So it's really using that information to turn back into making a high quality product. We do the same thing when we talk about engineering cells. I was saying we moved up to be able to build these automated platforms for engineering. And we do the same thing by taking machine learning loops to understand variability in guide, variability in nuclease as we evolve a protein, and variability around different parameters that render cells more or less susceptible to the transduction process. So it's really kind of an internal tool set that lets us build and optimize out. Again, it's taking engineering principles and reducing the complexity in biology. But we also use that in other ways, particularly around target discovery. one of the really interesting things we've been able to do is to edit the HLA locus, either class 1 or class 2, so that we have a resident peptide epitope embedded genetically in the construct. And so what that lets us do is build a repertoire library of putative epitopes that are all embedded in an HLA molecule. And so we can do that at very high efficiency. Obviously, using genetic tools, we can get a lot of redundancy in terms of the epitopes that are being presented there. So now we can start to take like neoantigen predictions that come computationally, derived from genome sequencing of tumor cells, and say, here's a list of candidate epitopes that might be recognized. We've separately built a T-cell activation screening platform that can give us a quick read when a T-cell receptor hits an appropriate peptide epitope. And so now we're in the game of building both TCR repertoire libraries and HLA epitope libraries to understand specific TCR recognition elements and relate that back to what's seen clinically in TIL and other clinically relevant samples. So it's a really fascinating space to be able to make these reagents accurately and then to be able to use bioinformatics tools to sort of tease out biology around it. So I trust I've introduced Synthego well enough. I also know I came here with a posse of people, and I'm not seeing them in the room this morning. Everybody had too much fun last night. So I hope this is recorded.
Yeah, we'll make sure that they all see that.
Thank you.
Manos, you want to go next?
Yeah, sure. So my name is Emmanuel Medzakopian. People also know me as Manos. And I'm the VP of Research and Development at BitBio. And I've got a background in, before moving to BitBio, I was an academic, and I have a background in developmental biology, cell engineering, and genetic screening approaches. And as an academic, I use these approaches to identify novel therapeutic targets for neurodegenerative diseases. BitBio is an exciting company to be part of because what excited me about it was the challenge that they had when I first heard it. I said, that's quite a big challenge, which is to basically be able to produce every cell type possible for human cells for human therapy. And just the size of that challenge that grabbed me, so I wanted to learn more. So what is BitBio? BitBio is a synthetic biology company which provides human cells for human therapy, for human cell therapies. And it's also a platform company, and that means that the cells that we design and produce are to be used also for research, drug discovery, and finally also, of course, cell therapeutics. And one of the exciting technology that we have is the cell coding discovery process that we have in-house. And this allows us to basically look at the final cell type that we want to get to and work our way back and design a platform, design the platform in a way that we can reach that cell type and produce it at high reproducibility and scale. Reproducibility and scale are very two key factors that we look at BitBio because they are bottlenecks that need to be overcome for progress in the field. And yes, so the way that we do that is actually, and now I can come into the way that we use machine learning or are planning to use machine learning in our pipeline. We harness the power of cellular reprogramming, and for that we need to identify transcription factors that turn on the code in iPSC to turn them into the cell type of interest. And that happens very quickly, and you can get your cells at a very short period of time from the moment you turn on the code. And the way that we do that is we have our technology, OptiOx technology, which is the on button to turn on this code. Machine learning and AI comes in for prediction, the prediction of the transcription factors that are required to drive that code. And that's quite a big task because we use large omics data sets that require harnessing and understanding to reach to a list of transcription factors that is predicted to drive that cell type identity. And this is where you require machine learning. So prediction of the transcription factors that drive the code. And then on, we want to also include and expand on machine learning where we increase the reproducibility and scalability of the cell types that we produce. And we want to provide to the community, again, for research, drug discovery, and cell therapeutics. And you require machine learning because the size and scale of the production will require it to be done in a highly efficient way, and combining machine learning with imaging can really get us to where we want to be and provide the confidence that we need to scale up to a size that would provide cells to a large population of users or patients. And yeah, I think with this, I kind of wrap up why I've jumped into industry through BitBio, because I basically run that platform in BitBio, the cell coding and identity platform that we have. And the challenge is quite large, and I took on that challenge. So this is why I'm excited to be in the field at the moment. So thank you.
Yeah, thank you. Keith.
Good morning, everyone. My name is Keith James. I'm the head of discovery at in-vitro, a Bay Area-based biotech company. I have a multi-decade career, let's say, in drug discovery. Most of that was spent at Pfizer, but I was also the president of a research institute here in San Diego for a privately owned Swiss headquartered pharmaceutical company called Ferring. I joined in-vitro about 3.5 years ago to build a machine learning enabled drug discovery capability at the company. So in-vitro was founded just over 4 years ago by Daphne Koller, a former Stanford professor and world-leading machine learning pioneer. And it was founded on the belief that the application of machine learning to biological data generated at scale would lead to new biological insights and new therapeutic opportunities. So the company was about 40 people when I joined. We're almost 200 now. But about 50 of those people are machine learning scientists, engineers, and data scientists. So the company's had an unusual build in incorporating machine learning right from the inception. So our approach is to build iPSC-derived human disease models and to characterize the phenotypes of these models using machine learning. And we study the phenotypes using high content readouts. That could be imaging, looking at morphological features of the cells, also transcriptomics, and also functional readouts where we can. And I suppose we as humans have evolved to be very good at recognizing some things like faces, but we've not evolved to be very good at interpreting images looking down microscopes with lots of colors and blurs and shapes. But it's possible to train an algorithm to be able to understand the difference between normal healthy systems and the diseased systems that we've created. So we have investments in neuroscience and metabolic disease, and the company's technology platform has matured now to the point where we can generate these cellular disease models. We can characterize their phenotypes, and we can perturb the models using pooled genetic screens, and then look at the consequence of those genetic knockouts. We use CRISPR technology to do that, and identify genetic perturbations that revert the disease phenotype back in the direction of the healthy system, or all the way back to the healthy system. And this has now enabled us to identify genes, hitherto unsuspected, that are clearly driving the pathology of diseases. And these will now be the basis of both small molecule drug discovery programs and probably oligotherapeutic drug discovery programs. And our platform is at the stage where we're now up and running with those kinds of programs. So the company's momentum is building, and our hope is that we'll be able to populate a therapeutic pipeline over the next several years.
Great, thank you. And last but not least, Isaac.
Hi, so thanks for the opportunity to be really excited to share this discussion. My name is Isaac Bentwich. I'm a medical doctor by training, and I'm the founder and CEO of Quris. Quris AI is a bio-AI company that focuses on safety prediction of drug candidates. We realized from the outset, and this led to the founding of the company, that the theme of this workshop here is really important. In other words, that machine learning and advanced biology are evolving impressively separately. And further integration between these two disciplines can be of help. Of course, this is all being done. We identified within this field the area of safety prediction as an area that where we can perhaps contribute to the important work that others are doing. The rationale being that there are strong processes moving forward on discovery using various methodologies. But at the end of the day, if you build a powerful discovery engine, be it more from immunology or from chemistry or from AI, at the end of the day, you have to go back to the bench to see if it works, and then go to mice to have a better understanding or guess, will it work in humans? And that fails miserably. 89% of all drug candidates, true as of today, fail in clinical trials. Recently, as you may have are aware, the Senate has approved the FDA Modernization Act unanimously after having approved by the Congress several months ago. And so this is really backing up steam. The part of that act is the removing of the 84-year-old mandate that the FDA had of using animal studies as part of drug development. This is, by the end of the year, will be signed into law, and so a major change. But if not mice model, then what? Very good technologies are there, and we're humbly joining and contributing our bit, which is to more tightly integrate iPS three-dimensional organ-on-chip work on the one hand, which has been shown to provide better predictiveness than other older biologies, together with powerful machine learning and deep learning. This combination of what we call bio-AI, or this tight combination of advanced biological models together with AI is a long-term passion for me for 3 decades now, through 3 other companies that I've previously founded. One of these in the 2000, Rosetta Genomics, focused on discovering microRNA genes using this approach, and indeed discovered most of the microRNA genes in the world. We were successful because of this tight integration of high throughput biological screening together with machine learning. We're now applying that to drug safety. Our collaboration with New York Stem Cell Foundation, a leader in iPS, in stem cell automation, allows us to not just test thousands of drugs and construct their safety profile and train the machine learning to identify it, but to do so on potentially thousands of different patients, not just multiple interconnected organs on a chip or patients on a chip, but doing so on multiple different patients. So we hope with that we will be able to support and integrate and facilitate the work that others are doing.
Wonderful, thank you. So obviously there's a lot of different applications of machine learning as it intersects with cell and gene technology. And you heard about some of the innovations that are currently being used by companies in this space. In our preparatory discussions, one of the things we talked a lot about is the development of disease models. So either patient-derived disease models for more precision medicine drug discovery, better understanding of mechanism of action of drugs, and then better patient stratification, kind of getting to the safety question and the scalability of drugs to target patient populations. And so, excuse me, I'd love to hear from the panelists a little bit about where they see the use of iPSC-derived patient models and the intersection of machine learning in the drug discovery space and the drug validation space, where we see that going and how that might change the way that we think about drug development, whether it's the testing for safety or the patient target identification. I don't know if anyone wants to pick that one up to begin, but I'd love to just kind of hear from the group about where you see that going.
I'll take the burden of being on your right side. we know. So I'll actually go outside the iPSC space with the first example. About 2 years ago, when the COVID pandemic had us all locked down, we were interested in part of a collaboration with an investigator at UCSF, a fellow named Nevin Krogan. And so Nevin has been studying how respiratory viruses interact with genes within the human cell that are necessary for viral propagation. And so he does that by doing immune precipitations of viral proteins and then understanding what human proteins they contact. And so he basically built a protein-protein interaction map where he mapped out all of the contact points, the number of human genes that touch COVID-based proteins. And so the objective there was to understand that structure-function relationship and look at repurposing drugs that could target the human protein and therefore block the COVID protein interaction, therefore block COVID infectivity. So he basically came to us with a panel of about 130 different human proteins for which he wanted knockout libraries. The premise being that if you knock the protein out, it should result in a concomitant drop in viral infectivity. And so we took 3 different cell lines that we knew were COVID infectable, and we also did the logical controls of knocking in or knocking out the ACE2 receptor. And so for the 133 genes, we made 3 guides per gene and then constructed knockout libraries in each of those 3 cell lines. And so having made those knockout libraries, we see very efficient knockout. We're in the 80 to 90% range for knocking out an individual gene. So the biology is going to read out fairly true for us in terms of infectivity. But we made those knockout libraries and then shipped them to investigators in Paris and New York and obviously at UCSF. And so from that, we saw about 10 hits that gave us more than an order of magnitude drop in COVID infectivity. And so we looked at those human proteins that whose knockout showed that result, and looked at a panel of repurposed drugs that had already been designed against those knockout opportunities, picked 3 of those, and drove them into early proof of concept studies. One of those, 10 months after we started, was in phase 3 registrational studies. So the reason it's exciting is that we made these 1,000 guides in 2 days, and one person took those guides, did the infectivity and shipped cells out, and we had the infectivity data back in 2 weeks. So this is like mind-boggling, being able to take a problem like that and to map it out, get some early hits on things that were linked, and be able to drive those rapidly down the drug development side. So I think there's just huge value in looking at CRISPR as a way to put diversity and use CRISPR as a way to bring diversity into target validation studies. So the second thing I'll talk about is really a setup for Keith. And so we have great interest in iPSC as a disease modeling platform. And we're particularly interested in rare and orphan diseases which can't get enough attention. And so our premise was that if we looked at a mutant allele linked to an orphan disease in a patient iPSC, in a patient background where all of the other allelic alterations or drift would have occurred, and compared that to the same mutant allele introduced into a healthy background, that by looking at transcriptomics, we would be able to deduce the secondary changes in the patient cell that really are the rheostats for disease. You have a driver disease, a gene that's responsible or linked to disease. It's interacting with a number of different pathways. Individuals that have disease have change in some of those pathways that make them either more sensitive to disease progression or make them refractory to disease progression. So these secondary alleles become a fingerprint that you can use and correlate against response to treatment. So you're starting to build in, based on these differences, hypotheses to build a gene panel for screening that you can then apply to the individual patient and ask, is he at risk for disease progression rapidly, or is this somebody that will respond quite immediately to disease? And so you realize within that there are a series of drugs behind the main drug that's out for treatment of disease that were not as successful. Many of those recognized allosteric variants of the original gene. And so you're starting to look at the ability to have diversity brought into a test background with iPSC and then go to a panel of structure function driven drug interactions to quickly come forward on the development side. So we really like the idea of building a genetic fingerprint that can be applied to patients in the rare disease space and used prognostically, and I trust I've set Keith up to speak to that. So the final thing I'll say there is that the value or need for this is very, very high. I was really fortunate to be at a meeting called RareX recently in San Diego that brought together a number of foundations that are participating in the rare disease space. So in other words, you have 50 rare disease alleles and you have 50 foundations that are trying to chase down cause and effect and to build alternative treatments for disease. These people need exactly what we're describing here, is access to a common database where these principles of dissecting out the omics around the individual disease have a defined development path. People know how to exercise and use that data and move quickly in the rare and disease space to do something about it. So it's something that I'm obviously real excited about, and I think more of that will come out as the panel continues, and I hope I've not talked too long.
No, it's a very nice setup. I mean, I think we've talked about the time efficiencies, and then I think the applicability to rare disease, and I mean, you're all in the cell modeling space, and so Keith, I'd love to get your thoughts on what Bob was just saying, particularly in rare disease.
Yeah, forgive me.
Yes. Yeah, so a number of sort of very attractive aspects of working with iPS cell-derived disease models. Obviously, you can study human tissue. If you're studying neurological conditions, then it's difficult to get access to the brain in a live person, but you can build a cellular model that replicates many of the features of neuronal disease, and you can perturb them in the lab and look at the consequences. So that was one of the aspects of this field that really attracted me, having spent decades working in programs that used highly contrived animal models of disease, and I'm sure you're aware of the many shortcomings of that approach. So we have found the ability to to create a cellular model that exhibits the sort of the phenotype of the disease, and then to study it using very high content readout technologies to be a very powerful way of building a much deeper understanding of what is driving the disease and what might be opportunities to perturb it. The challenges that we have encountered are to be able to scale the creation of these models. Obviously, creating a reproducible cellular model at scale that avoids the batch effects that confound machine learning approaches, is one of the challenges that we've grappled with. And also, as exciting as iPS cell-derived systems are, then they don't completely recapitulate, a primary cell. So we have an interest in liver disease, and we know that iPS cell-derived hepatocytes don't fully reach the level of maturity of primary hepatocytes, particularly, for example, with expression of serochrome P450 enzymes. So it's important that we also recognize the limitations of the technology and some of the challenges that have to be overcome in using them at the scale that is really required these days.
Mm-hmm. So you mentioned one of the issues for stem cell therapy is the kind of the inefficiency in development of a really relevant cell type. And Manas, you talked about BitBio really focused on identifying key transcription factors. I was curious how you see machine learning helping us really improve the directed differentiation of stem cells to make better models, but then also potentially better therapies.
Yeah, these are very excellent talking points and are very current. And at BitBio, the way that we are looking at this is we would like to combine the cell types that are disease relevant that we produce, and engineer the mutations that the field wants to study in these cell lines. We find that with our reprogramming technologies, we have minimized batch-to-batch, batch-to-batch differences in noise. So for us that's a huge milestone. And then we want to take it even further, where we want to combine different cell types that would be interacting in a disease, so making the model more complex. So now you can imagine a situation where you have high reproducibility in deriving the cell types, and then you are culturing them in a very reproducible way, so to be together in the dish, so you can study them. Now, going back to the machine learning attribute, so it's very important that the cells that you're studying in vitro are very, as close as possible or as relevant as possible to disease, and the cells that are implicated in disease, and maybe even the surrounding cells that they interact with. And the machine learning can really help us, hit home the cell identity, but also the cell state. Situation, cell functionality. So you can now start designing the cell to be as close as you like to the situation you're actually studying in disease. And where machine learning comes in is that you can use these high learning models where you feed in the information that either you produce specifically for the problem that you're tackling, and also whatever information is out there around the disease. Cell types, we have vast information at the moment that we can harness from genomic DNA sequencing, transcriptomic, and all sorts epigenetic data sets, combining that with specific data sets that are relevant to cell state and cell functionality. That's where machine learning would come in and actually bring that together to help us design the model to be as close as possible to what the field needs.
Mm-hmm. Yeah, that's great. So obviously there's a lot of innovation that will go into that.
Absolutely.
So, you know, we've talked a lot about the ways in which cell technology in particular can be used and combined to understand new questions, answer new questions. And, you know, Isaac, you talked about your interest in safety. And so I was, you know, curious to kind of start with you thinking about, there's obviously safety issues in the current generation of cell and gene therapy technologies. There's issues in targeting, you know, efficiencies, et cetera. And so how do you see your work and your focus in terms of iPSC organoids being useful in evaluating, for instance, the safety of a CRISPR, you know, in vivo CRISPR gene editing
Sure.
medicine?
So first, connecting it to what has been discussed, I think an interesting stepping stone that we're all at now is the evolution of more predictive biological models. iPSCs is one step in that direction. Three-dimensional miniaturized organs is a second step, not just a flat cell, a mono cell culture, but a co-culture of multiple cell types. And then interconnecting multiple such organs together gives a better predictivity, as has been shown quite dramatically over the past 18 months. And so, to your question, in the cell and gene area, this brings a question. These models of multi-interconnected miniaturized organoids and organ-on-chip devices have been shown to be more predictive. Are they accessible to cell and gene therapy? And so the good news is that this is something that's up and coming and has already been shown to be valid. Adeno-associated virus and other vectors have been shown over the past months to be effective in transfecting in organoids, especially if they're not too big in dimension. And so for us specifically, we've started with small molecules, which is easier to handle. But in selecting the models that we're using, as an example, focusing on organoids that are much smaller than other conventional models, so that cell and gene therapy will be effective in working with these models. So this has been an important consideration for us in a previous life. As I mentioned, I'm a short RNA guy, and so the whole notion of working with gene editing and cell and gene therapies is very dear to my heart, and I think that a lot of the bang will come from integrating machine learning together with these new modalities.
Mm-hmm.
Perhaps I can just briefly add to Isaac's comments. Another big attraction of this constellation of technologies is the ability to do unbiased experiments. So, you know, to create a cell model, to perturb it with a very large pool of gene knockouts, to look at the consequences using high content readouts like morphological change, for example. So there's no hypothesis driving that. It's really a very broad survey to understand the consequence of perturbing the system. So that we feel is very powerful, and it's the integration of this whole set of technologies that allows you to conduct that kind of experiment, which in principle could be literally a genome-wide perturbation of a model system, revealing sort of completely unexpected biology. I think that's very exciting.
Yeah. So you mentioned, you know, unbiased decision making in models. You talked about kind of the amount of information we can process with machine learning. In the Alliance for AI in Healthcare, one of the challenges we talk about a lot is the lack of data. So either the lack of curated data that has the bias removed from it so that the decision-making process is completely unaffected, and then also just the paucity of overall data for which we can make decisions on. And so looking a little bit in the future and speaking to the audience at ARM and at the AH, you know, how do you how do you see solving the data question from a regulatory perspective, from a technology perspective? How will you generate more of that data yourselves? And then what do you guys do to have confidence that the data you're generating is unbiased so that the machine learning algorithms can make an informed decision that you then don't have to second guess down the line?
Yeah, so from my point of view, there's also we need to educate each other on what type of data sets are really required, so that efforts are put together, consolidated for specific purposes. You can take different fields for their own different needs that start consolidating. For example, in Parkinson's disease, there is going beyond the iPSC models and so on. There is also a need to start collecting data, day-to-day behavioral changes, so that then that can feed into the physiology. And these data points, if you're collecting behavioral data sets over time, then you start ending up from multiple individuals, ending up with a huge data set where machine learning comes in and starts picking that apart. And then you add in the information, the information that's required from biosamples, and then moving on to the disease model. So it's when these things come together, that's where the power of machine learning can be really harnessed. So there needs to be a consolidated effort in each individual field, I think, to start thinking of, okay, what is required and where can we get to. And also a very important thing to think about, which is it's just been mentioned here, that the noise in the data can actually be problematic to really take advantage of artificial intelligence, machine learning. And one way that we at BitBio think of that is to set a standard. So if you have a reproducible model that day in, day out gives you the same metrics and are used alongside experimentations, that can give you a kind of a ground truth idea of where things stand, which hopefully would reduce noise. And then having that ground truth information in there may help actually clean up background noise data that can hinder machine learning. So this is one way that we're thinking about it. So yeah, setting a standard, and especially data sets that are coming between labs, they can somehow be different. If you set standards across labs, across experimentation, and then find an intelligent way to feed that into machine learning, it should help progress.
I can add briefly. We see access to data at the scale and quality that you really need to make the most of machine learning technologies as a huge challenge for the field, because of these issues with kind of compilation of data from different sources, confounding machine learning algorithms. So one solution obviously is to create the data yourself, where you have complete control over quality and scale. And that's what we have done. We've built literally a biological data factory to produce the data that we think is relevant. It's a very expensive way to go, obviously a huge investment. But we've also tried to avail ourselves of public domain data. For example, we make extensive use of data in the UK Biobank. As a Brit, it makes me very proud that the UK has provided this resource to the world's researchers, and it's only going to get better, and clearly other countries are beginning to move in this direction as well. So that's a very powerful source of deep phenotypic and genomic data on human cohorts that is definitely advancing the field, and hopefully there will be more of that. Consortia are another potential way to solve the problem, as Mams alluded to, where organizations pool their data, having agreed protocols for how it should be generated. So I think all of these are potential ways forward to tackle this challenge.
Yeah.
I'd like to add to what you're saying, Keith. Generating data obviously is an important part of the roadmap and really part of the exciting collision between machine learning and biology is this dialogue about the data, the predictiveness of data and generating data. And part of the future challenge that we see is in taking the next step or a next step of interconnecting multiple organs, not just tissues, organs. Because if we want to go beyond mice, there's a reason why we used mice in the first place, and it was, let's try and see the systemic effect of a drug, ADME and all of that. And so steps in this direction of interconnecting multiple human three-dimensional tissues is really important. It has already shown a better predictiveness, but very difficult to do on scale and reduce noise. That's where the integration between machine learning and specific in-house engineering that allows us to create these slowly, baby steps, creating these multiple interconnected three-dimensional organs become a reality. Working with these systems on a regular basis now, we see the tremendous power of low-level integration between machine learning and the engineering. Having the machine learning analyze raw data at the time of sampling, allowing it to be an interactive, dynamic system is very powerful. Like, I agree with what you said, Keith. It's an expensive proposition, but it holds great promise and is showing results. And as an example, rare diseases were mentioned before. Our focus, our first drug at Quris is for Fragile X, and within short months we've been able to progress a drug, the discovery of which began in academia and the development of which we undertook because there is no mouse model for Fragile X. So an extreme case of where you really need the simulation of biological simulation of the body as a system, and this drug is now preparing to move into clinical trials.
Wonderful. So before we move to the Q&A, I just had one. We've talked a lot about the high level, how the technologies intersect, kind of more the philosophical side. But one of the topics that came up in our talk is that these are multidisciplinary teams with very niche expertise in machine learning, and then the application to biology, and then biologists who are willing to integrate with more kind of digital technologies. And so, you all oversee multidisciplinary teams, and I'd love to hear how you've brought these teams together, kind of what are the challenges there, and then more holistically, what do we need to focus on for the development of a workforce that will really kind of thrive in this world?
Yeah, it's kind of, you're asking about how we bring cats and dogs together as
Yes.
a happy family. And I think one of the interesting things for us is looking at how engineers approach root cause and critical attributes of a product in development as opposed to the biologist. And so being able to manage that intersection so the reciprocal team understands what is critical from the opposing point of view has been a task for us. So one of the things we've done is build a homemade electroporator to be able to more efficiently gene edit cells, and the engineers went about building it without input from the biologist. First one through resulted in a major explosion in our factory because they hadn't understood the effect of the biology and the cells being added into the solution that was being tested. So we've just had some fun turns on how differently people would look at a common problem and how to try to unite them. I think for us going forward, it's about there aren't enough people out there to do this work in the computational space, and they don't come from the standard biology graduate programs that we're used to building. So I think what we're doing is looking out more towards people with engineering backgrounds or programs that have been heavy on the engineering side to find biologists that have gone through that have that kind of hybrid character. So I think we're, on top of that, we all know everybody wants to work remote these days, and I think we've also all figured out we can do that. I mean, I think in the gene medicine space, you can almost contract your data set to build your gene-linked hypothesis. You can outsource the kind of work that I just described from Synthego to be able to validate your target, and you can outside contract the ability to look at process development to make the therapeutic. I think if we can work with the FDA to take preclinical modeling out of gene therapeutics and the new gene medicines, you know, we're now starting to build a stream that can move much more quickly. So I think starting to look at regulatory change, the types of people that we're drawing into this environment, how they work, and trying to model the future a little bit is a lot of fun. So I'm going to pass it. You know, you're next to me and you got it.
No, happy to. It's a great question and topic to discuss. I mean, in our cell identity discovery platform that we have, it's quite huge in the sense of the requirement of multiple interdisciplinary functions that need to come together and orchestrate the progress of information across this platform until you get to the cell type of interest. So we embrace the differences that these different functions and the people behind these functions bring. The most important thing in BitBio is that we all work according to the values that that we have in the company, and it's the values that bring us together. And of course, there are difficulties in bringing different people and different points of view together. But when I say we've embraced it, is we always look for ways to tackle any misalignment. And I must say, up to the point, that's actually bear a lot of fruit. And embracing this diversity and this difference in mindset is actually fantastic and very invigorating. It keeps you on your toes, which is great. Yeah.
Yeah, it's a really important point. Since in-vitro is based in the San Francisco Bay Area, we compete with Google and Facebook and Tesla for talented machine learning scientists and engineers. So part of the solution is that we're looking for individuals who want to apply their capabilities to solving important human challenges, discovering new medicines, and there are many people who find that highly motivating, even though they could work on social media platforms or autonomous vehicles. The other thing that we've got going for us is the company, as I mentioned, was built from the ground up as a machine learning based organization. We have a charismatic CEO who I think is a magnet for really talented machine learning scientists and engineers from literally across the globe, which is a huge aid to us. And we're tackling very difficult, complex problems. I think that's also highly motivating for people coming from these computer science, machine learning domains. It's a struggle, but we have made a lot of progress. With regard to integrating biologists and machine learning scientists, then we used to talk about trying to create a bilingual workforce where people could understand each other. These days it's more of a multilingual challenge as we bring more and more disciplines to bear. But once again, there are more and more biologists joining the workforce who have had some exposure to machine learning and have some of the terminology and the basic concepts that helps them kind of build that bridge to their colleagues. That's been a help.
To continue on this, just to share with you the flavor of computer guys working together with biologists. 15 years ago, when I first ventured into this interesting area, I used to tell this story. You go to your AI guy and say, listen, for tomorrow, we need to change something. I want the sky to be down and the land to be up. And he thinks for a moment and says, sky, soil, up, down, one parameter, no problem. You go to a biologist and you say, for tomorrow, I need your test tube to move from this place on your bench to this. He said, what are you kidding me? So, you know, this, and it's not because of narrow-mindedness. This is the reality. It is difficult. These are very different mentalities. At the same time, the synergy between them, especially when this is intimate synergy of working together on at the raw data level, at the design level, is tremendous. Israel is also, Tel Aviv houses AI teams of some of the world's leading global corporations. We compete with them very successfully in attracting top talent. Because you know, how long can you make stock holders richer and do another app that's a bit faster than something else, rather than changing the world, doing something that's really interesting. So there's a lot of fascination there, and a new generation is growing up now within this synergy of people, like you mentioned, that computer guys that have some exposure to biology and vice versa. Exciting time.
Wonderful, thank you. I think we have time for a few questions, and I know they're going to hand out a box that is also a microphone.
I don't need this because Mom told me God gave you 2 ears and 1 mouth. Learn to use in proportion, and that's never worked. A great talk. Thank you. I applaud you. Some dumb questions from a biologist, hardcore, blood and guts, basic pharmacology biologist. So you'd mentioned, Robert, secondary allelic variants and how that's a predictor of disease progression potential and possibly response. Should we start thinking about, and are you guys thinking about, using that type of system with AI as a proactive stratification strategy for clinical trial development? Because I've worked on cancer therapeutics all my life, and I've done everything from discovery out to ODAC submissions for approvals, and failed in some and done okay in others. The question is that allelic variant, secondary allelic variant story is very intriguing, and is that something that could be used instead of, you know, we stratify for age, we stratify for this or that or disease stage? Would this be an additional tool to add to the armamentarium?
Short answer, yes. And
good.
Yes.
Okay, good. Next question, and then I'll hand this thing off. I warned you guys. You were warned. Risk-benefit. Risk-benefit and target validation are inextricably interlinked. You can identify a novel target, but if the person's head falls off, it's not a target anymore. So, and it's also relativistic based on the disease type. Chronic diseases that are non-life-threatening imminently have a lower tolerance for safety. Acute diseases, stage 4 metastatic melanoma, we're much more tolerant of toxicity. We used to kill 2 out of every 50 people, out of every 100 people we treated with 5-FU in colorectal cancer at MD Anderson when I was a kid, going to clinical grand rounds. So how does AI get built into that rheostat of safety tolerance for different disease categories? And now I'll give this thing to Jacob. I got a bunch of questions.
Briefly, part of the reason that we've decided to focus on safety first, the platform we're building is good for efficacy as well. We made a strategic decision to focus on safety, because from an AI perspective, as was mentioned earlier, rather than developing a gazillion disease models, it's one disease model that you focus on and that can co-train based on drugs in different modalities. Absolutely, the ability to better stratify patients, and in a sense, especially when as systems like the ones I'm mentioning of multiple interconnected organs are studied by AI with real-time nanosensing, etc., you can in effect begin running clinical trials on a chip. being able to run before the clinical trial with the IPSs of the patients that you would have selected, with several models of organs of these patients. It's not an end-all be-all solution. It's a long and painful journey. We're just in the baby steps of it, but it is extremely exciting because you can, in fact, imagine, start from very low, even phase 1. Wouldn't you like to run your phase 1 before you run your phase 1 in a lab? How long will we be testing drugs on humans before testing them on humans in a lab? So these are exciting times.
Do we have time for a couple more questions?
Yes, a question here on basically technical capability and acceptability from a regulatory perspective, right? I think we have great advantages here in what we can do technologically. We have belief, specifically in companies on gene therapy about what these iPS models and other organoid models can bring. At the same time, a high level of frustration on, the inapplicability or unavailability of reliable animal models, and then the response of regulatory agencies to kind of bridge that gap. What is your collective experience in terms of that dialogue? I think Bob mentioned some of that. And what do you see as a trajectory to get more acceptance for these types of approaches and not kind of bang our heads against the wall for trying to find an animal model that is pretty elusive?
So, I just wanted to say, I mean, it's like a question for the question as well. It's from the regulatory part of the question is what triggered this thought. It's also what other bottlenecks for machine learnings will we have in the near future? So now we're discussing about how to provide the vast amount of data sets that machine learning requires. But once that's done and you start solving very complex problems and there's an output coming from artificial intelligence, from a regulatory point of view, you may be asked to actually untangle all that analysis that happened that gave you that output so that you can understand where it came from. That may be a future, bottleneck that we should. I'm sure the field is thinking about this. So, that's one thing that we need to consider beyond the usual issues that we go through. Yeah. So that's what.
Yeah, that's great.
I think we have time for one more question.
Well, then I lied. Allometric modeling to get to drug therapeutic exposure and using AI for that. I mean, biotransformation, tissue residence, half-life in different organ systems is critical for predicting response. We had great drugs we used, both biologics and small molecules. Cell and gene therapy, a little bit different, but basic principles of pharmacology apply. The therapeutic target has to have a certain level of volume or quantity and duration of exposure to the therapeutic. AI would be custom tailored to that because the variabilities when you try to do classical ADME and the data and generate from that an assessment of, we're probably going to be able to get to a dose in the clinic that works, has been weak for the last 50 years. I know. I've tried using nude mouse systems like that and other systems. Please fix that.
Yeah, I've no doubt that if we could access sufficient training data, it would be possible to have an algorithm learn the relationship between molecular structure and pharmacokinetic properties. I think accessing the data to train that model is the big stumbling block at the moment.
Yeah, I just have to throw one other point to Alex's question, and that was last year there was a guidance document from the FDA on use of antisense oligos, and they basically prescribe eliminating the long-term preclinical toxicity studies, acknowledging the value in animals of acute toxicity, but arguing that because of the lack of concordance between human and mouse, you don't gain fruitful data by running a preclinical in vivo safety model. So you should go back and read it and see the direction I think the FDA is going is very positive for the space.
That's good. So unfortunately, we are out of time, but appreciate you all showing up this morning. Give a round of applause for the wonderful panelists sharing their insights and expertise.